Good evening. My name is Clara Gaff and I'll be your MC for tonight. I'd like to begin by acknowledging the traditional owners and custodians on the land which we meet today, the Kulin Nations, and pay my respects to their elders past and present, as well as elders from other communities who may be present here today. So welcome to our public forum tonight. This is hosted by the Walter and Eliza Hall Institute. It's part of the Walter and Eliza Hall Institute's Science in the Square Festival and part of the larger celebrations of the Institute's 100th anniversary, making it the oldest medical research institute in Australia. It's been a year of celebration. Tonight, we're posing the question, will there ever be a cure for cancer? We're fortunate to have 4 leading Melbourne cancer researchers and clinicians here to answer that question, we hope, and to discuss further. During tonight's event, we're going to have each of the 4 speakers speaking briefly, and then there'll be about 45 minutes for questions at the end. So I'm going to ask you to hold your questions until that discussion, and we'll have some mics floating around. Also, tonight's event is going to be recorded and live tweeting will be used with the hashtag that's up there, #WeHi100. So many of you here tonight will know somebody who's had cancer. Unfortunately, 1 in 2 Australian men and 1 in 3 Australian women will be diagnosed with cancer by the age of 85 years. So within the next 5 years, it's expected that about 150,000 Australians, that's 500 times the capacity of this room, will be diagnosed with cancer. Even though cancer is the second most common cause of death in Australia, the good news is that survival is increasing, with almost 2 in 3 people now surviving 5 years past their diagnosis. We're also seeing improvements in the treatment of some cancers, and our speakers tonight will give you some examples of those new treatments and new approaches. So we're seeing now a move towards a more personalised approach to healthcare and cancer treatment. Clinicians are using information from a patient's cancer genome to help them make decisions about the treatment and care of that person. The speakers today are all from leading organisations in Melbourne who are working together through a number of initiatives. One of those is the Melbourne Genomic Health Alliance, which I am the director of, which is an organisation or an alliance of 10 leading institutes and hospitals that are really working at putting genomics into healthcare practice, and that's broadly in cancer as well as inherited conditions. The other leading initiative is the Victorian Comprehensive Cancer Centre, the VCCC, which was formed in 2009, and it is working to really drive improvements in the cancer care and availability of new treatments and detection of cancer. So rather than this sort of brief overview and introduction to cancer and the complexities of collaboration, I think we really need to turn to our speakers to hear in much more detail their research and care. So the first speaker is Dr. Kate Sutherland. Kate is a lung cancer researcher at the Walter and Eliza Hall Institute. During her PhD, Kate studied breast cancer before she turned her focus to lung cancer. In the past year, she's worked at the Netherlands Cancer Institute, where she made important findings about the cells in which lung cancers originate. Kate has returned to the Walter and Eliza Hall Institute and is now a senior researcher where she's focusing on developing models of lung cancer that can be used to identify and test new therapies for cancer patients. Please welcome Kate.
Thanks, Clara. Can you hear me? Okay, so tonight I thought I would give you all a little background in what cancer is and how cancers can arise in our bodies in different tissues and organs. And then I'd like to touch upon the research my laboratory is taking on making new models of lung cancer and using these models, as Clara said, to identify new and clever ways in which we can combat this really devastating disease. So, as you are, I'm sure, very well aware, our bodies are made up of billions and billions of tiny little cells. And there are many different types of cells in our body, and each of these types of cells have a specialised job to perform. So lung cells, they are involved in gas exchange, while gut cells are involved in absorbing nutrients. In contrast, we have immune cells in our body, and these cells help defend our body against invaders. So the instructions on how we build every cell in our body is encoded in our DNA. And our DNA consists of chemical letters, and that's what is shown on the right-hand side of this slide. So we have 4 letters, T, A, G, and C. And combinations of these letters form genes or different words. And these words are packaged into chromosomes or books, and these are found in every individual cell in our body. And it is really the combinations of different genes that make a lung cell, and a skin cell, a skin cell. But what about a cancer cell? How is that cell different from a normal cell in our body? So cancer cells have DNA mutations. So what do I mean by this? Well, the DNA in a cancer cell has undergone an alteration. And that's what I've shown here by this lightning bolt on the right-hand side. So how can these damages or alterations occur in our DNA? Well, as you can imagine, if you smoke cigarettes, you can get damage to your lung cells, or if you're exposed to UV sunlight, you can get mutations in your skin cells. But how does this lead to cancer? Well, from this little cartoon on this slide, you'll see that on the left-hand side, here, if you think of our cell as a car, then some of our genes actually encode what we would call accelerators. So these genes tell a cell whether they can grow. So at a go signal, the cell knows, okay, I can grow and form a new cell. But if a mutation occurs in this kind of gene, in this accelerator gene, then actually now the cell doesn't know when to go and when to stop. So it really just goes all the time. So this is how cancer can develop. And maybe this is best represented on this slide, where cancer cells are different from normal cells because they have their foot on the accelerator and they are dividing and growing too much. So if we look at this red cell, which has an acquired mutation or a damage in one of these growth promoting cells, then the cell divides uncontrollably, so it doesn't know when to stop. And this forms a group of cancer cells that we call a tumour. And as tumours progress, some of these cancer cells can travel to other parts of our body and then form new tumours in these distant parts. And this is what we refer to as metastases. So cancers are really cells that have undergone changes and that divide uncontrollably in different tissues of our body. So there are many features that make a cancer cell, and what I've shown on this slide here is that cancer cells can undergo many changes that make them different from normal cells. And it's these features that we call hallmarks of cancer. And tonight we will, our three other speakers will discuss various aspects of these hallmarks and how we can use these hallmarks to then stop the cancer from growing and taking over. So now I'll just touch upon lung cancer. So as Clara mentioned, I've been studying lung cancer now for about 10 years. And really from this slide, I just wanted to point out that lung cancer is actually not just one disease. So in fact, there are 4 different kinds of lung cancer. And if you look at these pictures on the slide, you can see that these different types of lung cancer look really different. So we can't classify lung cancer as one disease. So then we can't use one treatment, one-size-fits-all treatment strategy to really target this disease. So in order to treat lung cancer better, we need to devise clever ways in which we can target specifically each of these different types of tumours in the body. So as a lung cancer researcher, we are interested in really what wires cancer cells. And what I mean by this is I tried to depict in this slide here. So we're all familiar with mobile phones, and I'm not at all technically minded, but I can imagine that the circuits that wire an iPhone and hold all the information to detect calls, to send text messages, to hold photos, etc., is quite complex. And this complexity is actually the same with an individual cancer cell. So an iPhone has a certain wiring system, and a cancer cell of one particular type has a certain network of interactions that really make it that type of cancer cell. But then you've got an old school phone, and this is the kind of phone that I give my young children to play with, and that has a different wiring system and a different network to the iPhone. So as you could imagine, this could be a tumour in a different individual. And if you look on the right-hand side, you can see that the network of circles and lines in this cancer cell is different to the previous slide here. So this is really just trying to highlight the need to look at individual patients and their tumours, and then define specific therapies that really target that individual's tumour. So my lab is interested in teasing out these complex networks to really find out which are the important networks that make the cancer cell grow and divide and metastasise or move to other parts of the body. And we can do this by looking at cancer cells in an isolated culture system, like on the left-hand side, but this really doesn't mimic how we know the cancer cells interact in human tissue. So we are focused on developing complex model systems whereby we can study the cancer cells in the environment that mimics the human disease. And we know that these cancer cells really interact with many other different types of cells in that particular tissue. So we really want to understand these networks. And we really use these models to address important biological questions, such as which cancer mutations or changes are most important, and in which normal lung cells do these cancers originate from. And really by addressing these questions, we can exploit the features of the cancer cells that are important in order to design better treatments. So tonight, hopefully we'll be able to show you that by exploiting these different hallmarks or features of cancer cells, we are able to design better weapons or better targets to fight specific cancers. And it's really this in-depth understanding of how cancers work in a biological setting that really enables us to design new and clever ways to treat cancer. So I'll just highlight that Kylie Mason will talk about how we can kill cancer cells, and Grant McArthur will talk about how we can harness the immune system in our bodies to now attack the cancer cells. And I'd like to leave it there. Thank you.
Thank you, Kate. I think we can starting to see just some of those complexities within cancer that make cure a real challenge. The next speaker is Jayesh Desai, Dr. Jayesh Desai, a medical oncologist at the Royal Melbourne Hospital and the Peter MacCallum Cancer Centre. He's also a clinical research fellow at the Walter and Eliza Hall Institute. Jayesh's clinical and research interests are in the development of new anti-cancer therapies and how we can effectively bring these into clinical trials programs. Please welcome Jayesh.
Great, thank you very much, Clara. And I think my talk really segues from what Kate had introduced in terms of, understanding the complexities of cancer biology. And, for us as clinicians, how do we try and work with our researcher colleagues to bring this into the clinic and hopefully develop better treatments for our patients with cancer? So, as we've heard, we need to understand that cancer is actually a complex, really a set of diseases. It's not one disease. And the progress that we will make and the scientific tools like the ones that Kate has talked about are really going to help us to try and understand at a greater depth how cancer as its makeup of diseases actually behaves and what makes it operate and what makes it function and so on. And really, in taking that a step further, how we can actually determine those changes in an individual patient with cancer. How can we actually do that, and how can we bring that into developing better treatments? So if we sort of, there's this concept of personalised cancer medicine that I will spend some time talking about. And really what this is about, until probably the last decade or so, when we looked at someone with cancer, we really thought of this as being a somewhat homogenous disease. So let's imagine this was a group of patients who all were affected by bowel cancer. And when we saw these patients, we knew that they would behave differently, they would respond differently to different therapies. But the reality is we didn't have the tools at that time to really understand why it was that these patients, who all, for all intents and purposes, had the same cancer type, actually behaved differently. Taking that a step further, what we've been able to do now is to actually try and break this down a little bit further. So we've been able to, using complex tools, molecular tools and genomic tools and so on that the others have touched on, break down cancer into its subtypes and really break them down into their subtypes based on the actual biology of those individual subtypes. Bringing that into the clinic requires a molecular test. It requires a specific test that we can do that not just tells us this is a bowel cancer, based on where it's come from, for example, but actually work out that this is a bowel cancer that has a particular driver, a particular feature that is making that cell grow and is therefore the thing that is making it cancerous and so on. So going back to this slide, so we think of cancers and think of the complexity of cancer, and this particular paper called The Hallmarks of Cancer was actually very important in trying to allow us to put that together. From a personalised perspective, this perspective of can we do a test and can we determine what's going on and can we use that to take things further, there are some hallmarks that we currently have the ability to do some tests for, and they include the ones that are highlighted here with these particular arrows. There are others that clearly are critical from a cancer point of view, but to some extent we have the ability to test for that and to understand biologically what's going on, but we're not quite there yet, and clearly a lot of our efforts are going to focus on not just the areas that are highlighted with the arrows, but also in some of these other key hallmarks as well. And the question really is, can we therefore take this personalised approach to it? Can we understand which hallmarks are controlling the signalling or the cancer process in an individual patient? So again, sort of using this circuitry analogy. So as perhaps not quite technophiles, Kate and I have obviously chosen to sort of go down this path, which says a lot. So let's imagine this is a normal cell. And in fact, cells behave much like an electric circuit. And a switch controls a normal cell, as we have here. That switch goes on, right? And that cell, therefore, is switched on and does what it's been designed to do. This is actually a fairly carefully controlled and regulated process under normal circumstances. What happens with cancers? So that switch, in fact, gets changed in some way. Kate talked about mutations, for example, in DNA, and a lot of these changes occur through DNA, and how that DNA interacts, or how it actually then feeds into the proteins that are these switches. And in some ways, we think of these switches as being many of those hallmarks, right? So a particular process that's now been changed in some fundamental way, and when that happens, this switch is now always on. There's no on-off button anymore. This switch is now on, much like a short circuit. So what can we do about that? Well, a lot of the therapies that have been developed over the last 10 to 15 years have tried to play on that fundamental process that occurs in cancer cells. Can we determine what that switch is? And by determining what that switch is, can we actually use a treatment, as is highlighted in the blue octagon here? Can we actually use a treatment to stop that switch from being on and essentially turn that switch off again? So therefore, this cancer cell stops doing the things that it has been doing. And that's really the basis for a lot of what are called targeted therapies. So taking that a step further, in fact, now there are many cancers that we are able to do a test of that switch, a molecular test as we call it, that is able to break down those cancers into a number of individual subtypes. So as an example here, in a patient who has colorectal cancer, we're able to do some fairly simple genomic testing, and with that, break it down into a number of what are called molecular drivers. So these particular genes that are changed in some way or the other, and they are the things that cause the short circuit now in that particular cancer cell. And in fact, this is now translated into a number of therapies that are able to stop those switches from doing the things that they're able to do. And this is a recent list of about 20-odd treatments for which we have a particular molecular test that determines what that gene is that's been switched on or changed in some critical way, and for which we have a drug that is able to stop that process from occurring. And these, of course, are what we know as targeted therapies, and this approach is known as personalised medicine. So to sort of really sum that up, what personalised cancer medicine really is, in a simplified way, is the ability to diagnose a cancer, but to now take it a step further and to actually analyse what that cancer's driver is. By knowing that, knowing what that target is, as we call it, can we actually use a treatment that is specific to that molecular target and therefore stop that process from occurring? And then, of course, in someone who's now being treated for their cancer, can we use some of these more sophisticated tools to monitor that patient's cancer progress and to understand what's going on in that more in-depth way? So how, in terms of how we translate this into practice, the critical thing here is this, really quite complex but in-depth collaboration that needs to occur with our scientists and with our clinicians and really with our healthcare systems as well. And as Clara had mentioned in the introduction, it's critically important that we actually work together in a very collaborative way for this, so that we can take some of the rapid advances that are made in technologies for testing, cancer genomics, for example, how we actually take that into developing new therapies, how we test those therapies in patients, and how we actually understand what's going on now in a more biological sense. It's critically important that we work together for that, and that's certainly what's going on now. So taking that a step further, what really, where are we at the moment, and what are the next steps here from that point of view? So certainly molecular targeted therapies, so these are, treatments that are linked to the molecular test, which is critically important, represent an important building block in treating cancer today, as I've shown. There are many treatments that are now available, and selectively these can be quite useful. They're quite useful at controlling disease, and they can do it very effectively in some people. And they can often do this with, manageable side effects for the most part. Certainly, if we want to generalise, more manageable side effects than what we might see with chemotherapy. And that's really where we are today. But as is often the case, it's not quite as simple as that. So one of the things that often happens, and for all intents and purposes eventually happens for many of the cancers that we treat with these targeted drugs, is that resistance will often develop. So here we go back to our circuit diagram analogy, and we have a treatment which is still effective at controlling that particular circuit, but what often happens is a second circuit or a sort of a bypass circuit develops whereby that light switch is now on again, and in fact that cancer cell is now able to grow. So there are resistance pathways that develop, and they develop with many of these targeted treatments over a period of time. So clearly understanding what those are and coming up with strategies to overcome those are going to be really important if we're wanting to develop really better targeted approaches for cancers like this. And the second thing, of course, that can happen is that some cancers are just too complex. So, this is a cell with a series of what are called molecular pathways here. And if we want to go back to the electric circuit analogy, the reality is that some cancers are just too complex, and that simple process of a single pathway with a single node and stopping that is just not going to be effective, because there are in fact many switches within some cancers. And clearly for those, we need to find other approaches, less targeted approaches as I call them, and perhaps in some instances better understanding functionally what goes on. So not just whether a switch is on or off, but whether that switch that has gone on is actually doing something. What are the key switches that are driving that particular cancer? And that's particularly important. So just to sum things up, I think the next steps in terms of taking this personalised approach to managing cancer, so that's clearly where things have gone in the last decade and represent a critically important part of what we are trying to do today, but they really represent some of what we need to be able to do if we really want to cure more people with cancer. So if we go back to our hallmarks, clearly we need to continue to refine our understanding of cancer biology and how we're able to test for that, better tests and better understanding of those tests. Perhaps incorporate other hallmarks, and you'll hear from my colleagues who will follow me about how we might want to think about other ways beyond just looking at these what are called signalling therapies. How can we look at other ways of also adding to that as well? Perhaps coming up with combination therapies, and that's certainly something we're looking at the moment, that target more than one hallmark so that those cancer cells don't have the ability to develop resistance or take a longer time to actually do that. So with that, I'd like to thank you.
Thank you, Jayesh. Our next speaker is Kylie Mason. Dr. Kylie Mason is a clinician at the Royal Melbourne Hospital. She's treating patients with diseases of the blood, including cancers such as leukaemia and lymphoma. Kylie is also a cancer researcher at the University of Melbourne. Her current research interests include manipulating the cell death pathway to treat blood cancers and studies on the blood clotting cell platelets. Kylie is going to talk more about a new class of anti-cancer drugs called BH3 mimetics, which are currently in clinical trials for treating blood cancers. Please welcome Kylie.
Thank you very much, Clara. So in my day-to-day life, it frustrates me that whilst we're able to treat and indeed cure many of our patients, particularly with blood cancers, we still have a large number of patients who do eventually die of their disease. And it's that that drives me towards finding better treatments for patients not only with blood cancers but cancer in general. So indeed, many of the advances in cancer medicine have been made in haematology, and haematology being the field of blood and blood cancers. In Melbourne, the treatment of acute lymphoblastic leukaemia, which is the commonest leukaemia in childhood, was revolutionised at the Royal Children's Hospital just down the road. And it was found that by combining multiple cancer drugs together, you circumvented some of the mutations that both Kate and Jayesh have talked about by targeting multiple pathways at once. At that time, and this was back in the 1970s and 1980s, they didn't have the detailed molecular understanding that Jayesh, for example, has just showed us with the detailed diagrams. But they knew that they needed to target more than one pathway because patients receiving just one chemotherapeutic agent would go on to have a response for a short period of time and then relapse. And essentially, it was like an escape of the cancer from the treatment that they were having. And so this really set the paradigm for modern cancer treatments with the treatment of ALL, or acute lymphoblastic leukemia, in children, and that's been followed through in multiple cancer therapies since then. One of the other dramatic breakthroughs has been in Hodgkin lymphoma, a type of lymphoma that's, I guess, most famous because Delta Goodrum and a few other famous people have had it. But again, it's a cancer that we've been able to cure the vast, vast majority of children and young adults with this disease. As an example of this, I can show you where some of these advances are often quite dramatic. So we used to talk about acute myeloid leukemia as just one disease, but as we've developed a more detailed understanding about the genetics and the genetic mutations, we've found a particular subtype called acute promyelocytic leukemia that has a distinct genetic mutation that causes the cells, just as Jayesh and Kate have spoken about, to be switched on all the time. So it's around 5% of all leukemias and affects all age groups from children through to the elderly. And in the 1970s, everybody with this disease died. In the 1980s, with this combination chemotherapy, it still had quite a poor outcome. And this was because patients who were treated with chemotherapy still died, particularly in the first week or two of therapy, of a severe bleeding disorder that occurred when these particular cells, the very large cells you can see there on the blood film, and the leukemic cells, released all of the gritty granules that you can see there, and they caused the blood to become very thin, and patients bled to death. It was a tragedy. When the leukemia was becoming under control, the patients would bleed to death. However, in the 1990s, it was discovered that all-trans-retinoic acid, or ATRA, which is really just a derivative of vitamin A, would cause these cells to mature, and they would no longer release the granules, and hence patients would be able to have the chemotherapy and survived, and the survival increased dramatically, so more than doubled. And these days we also use arsenic, often not thought of as a cancer treatment, but is in fact a very effective cancer treatment for this particular type of leukemia. And these developments have been made through a combination of clinical trials, but also laboratory research. However, some cancers are very difficult to cure. And we've talked a bit about how this is because they have many, many different mutations. And for example, one cancer may not be the same disease in everybody. One cancer that I deal a lot with is chronic lymphocytic leukemia, and this is the most common leukemia in the adult population, and has a very, very heterogeneous or varied clinical course, with some patients living with this disease for many, many years and never needing treatment, whilst others might need treatment within a few months of diagnosis. However, it is considered to be incurable. What is unique in some ways about CLL is that, as well as the cells growing at a faster rate, so always being switched on like many of the standard cancers, CLL cells often fail to die when they should. And anyone who's familiar with, for example, our skin cells will understand that our cells go through a natural process of life and death. They're born, they grow up, and they die. and their time when they die is pre-programmed through their genetic code. Some cells, such as our blood cells, die quite fast. In fact, our white cells only live for a couple of days, many of them in our blood systems, whilst other cells, for example our brain cells, are often with us for life. So these CLL cells that fail to die have that hallmark of cancer called evading death, as marked there. And we can see these CLL cells, and they're the purple cells in the blood there. And as they become more and more numerous, they just gradually overpopulate over time. And so over time, the other cells in the bone marrow, which is where the blood cells are normally grown, become overcrowded. And it's a lot like weeds in a flower garden. And if you don't actually kill the weeds, gradually they will just grow and suffocate essentially the normal cells that are growing there. So this in detail is controlled by proteins. And these proteins are encoded by the DNA that Kate talked about earlier. And there are proteins that are pro-life, so they support and they tell the cell to live, and there are proteins that tell a cell to die. And normally this is a very delicate balance in every cell. However, in cancer cells, what we often see is that the pro-life proteins significantly outweigh the pro-death proteins due to these mutations in the DNA. And they produce too many of the pro-life proteins. And as you can see represented here, the balance switches in favour of the cell living and not dying when it should. So one of the key things is understanding exactly what these mutations do, so that instead of targeting cells in a blanket way with standard chemotherapy, we can target this more specifically and target hopefully more of the cancer cells and less of the normal cells with cancer treatment. So the new anti-cancer drug called venetoclax that we've been working on and was developed through research done at the Walter and Eliza Hall Institute, but also clinical trials done in Australia as well as overseas, has shown that it can correct this balance. So it essentially mimics the normal pro-death proteins and corrects the balance in the cancer cell back to that of a normal cell and telling the cell to die when otherwise it would have lived. The specifics of this cell mean that it does target the cancer cell much more than the normal blood cells or the other cells in the body, resulting in not only treatment that hopefully is more effective, but also it has reduced side effects. So with venetoclax, the trials internationally have shown an excellent response. And you can see here a gentleman who has significant lymph nodes in his neck, and you can see from the swollen neck there with enlarged lymph nodes right around the neck. And that's showing us that the lymph nodes are all full of leukaemia, they're full of disease. And only 7 weeks after he commenced treatment with this particular drug, you can see the cells and the lymph nodes have shrunk. And indeed, with this drug and drugs in the similar group, as an example of this sort of more personalised medicine, we're able to see at times cells dying within hours when we look down the microscope, and we see shrinkage of lymph nodes within days, something that we didn't generally see with standard chemotherapy. So the exciting thing was of seeing many patients having a long-lasting responses with CLL and lymphoma with this class of drugs. However, as Jayesh has also mentioned, we're now starting to see, particularly as we're using this drug as a single agent in clinical trials, some escape mechanisms. Some cells, some patients where the CLL cells are now starting to evade this way of killing cells. And so we're having to look in more detailed fashion as to exactly why that is and in what people and what CLL patients this happens in. So one of the main benefits of chemotherapy that is targeted and more personalised and particularly targeted towards the cancer cell is the side effects of treatment are reduced. Not only, for example, in this situation, patients are able to take a tablet daily so they can get about their normal lives, continue going to work or school, and instead of having to be hooked up in hospital on chemotherapy through a drip. There's also less risk of the toxic side effects of chemotherapy, such as severe infections or vomiting and diarrhoea that are often associated with standard treatment. Another important feature is often these drugs don't cause hair loss, and hair loss, although hair does grow back, is a significant side effect of cancer treatment that often alienates patients with cancer and makes them feel somewhat more obvious with their cancer than they could, and stigmatises them in the workplace or in education. So the good news is that many people do survive their cancer. So the chance of surviving cancer in 5 years, and this is data collected by Cancer Australia, is that 67% of people who had a cancer diagnosis between 2007 and 2011 had survived at least 5 years from their original cancer diagnosis, so just over 2/3. And at the end of 2009, there were over 800,000 Australians, and probably approximately 1 million Australians now, who are alive and have been living with cancer, having been diagnosed somewhere within the last 28 years. So there are people who have been diagnosed with cancer, living with cancer, and many survivors of cancer throughout our population. And in fact, around 5% of people in their 40s, and this is data from the USA, are cancer survivors, and the data is very similar in Australia. So the goal is not only to improve cure, to actually effect a better cure. And that's really important because one of the major side effects of cancer treatment, although we know very much about the nausea, the vomiting, and the short-term side effects such as hair loss, not many people know about the long-term side effects of cancer treatment. And unfortunately, the long-term side effects of cancer treatment tend to be worse the younger you are when you have your first cancer treatment. And so almost all adult survivors of childhood cancers, so I spoke in my very first slide how we are now able to cure the vast majority of young children with leukaemia. The vast majority of those adult survivors of that treatment have a chronic health condition. And in fact, by the time they're in their mid-40s, more than 80% of them have either a life-threatening or a life-disabling condition. For adults, the rates of a secondary cancer or a secondary problem are a lot less, but still highly significant. Around 1/3 of people treated as an adult will in fact go on to have a risk of another cancer or a chronic health problem, which can be quite significant and result in long-term mortality or death or morbidity or illness. So one of the main keys to improving cancer treatment is not just focusing on cancer survival, and whilst that's obviously one of our ultimate goals, it's actually looking at improving toxicity. And by targeting the cancer cells directly, and I've given you one example tonight with the drugs in CLL, how that might be done, how actually reducing the damage to the normal tissues, enabling not only the replacement of standard chemotherapy drugs, but also the reduction of the dose of standard chemotherapy drugs, because a lot of the side effects of the standard chemotherapy drugs are related to the cumulative dose that people might have over their lifetime or over their original cancer treatment. So this not only reduces the risk of second cancers, it reduces the risk of long-term health problems, and that is exceedingly important, not just for the individual, but for the health budget as we go on to have so many more cancer survivors living productive lives. So treatments for curable cancers, and one of the big focuses for patients with haematological or blood cancers is now about looking at that patient, looking at the potential genetic mutations, picking a personalised treatment or a treatment that reduces this toxicity, not only the short and long-term toxicity, and trying to plan the treatment with the aim of providing the most effective treatment, but with avoiding short and long-term toxic side effects. Thank you.
Thank you, Kylie. Our final speaker tonight is Professor Grant McArthur. Grant is a consultant oncologist and cancer researcher at the MacCallum Cancer Centre and the inaugural Lorenzo Galli Fellow of the University of Melbourne. Grant's research interests include clinical trials of targeted immunotherapeutics, personalised medicine and melanoma. His research group investigate new targets for cancer treatment that control cell growth, division and differentiation. Thank you, Grant.
Thanks very much, Clara. It's fantastic to be here tonight to address what is almost like a question of our age, right? How can we get more cures for cancer? We're doing well, and I've got one take-home message. The way to get more cures for cancer is to invest in science. We've heard a lot of great science tonight. This is Science Week, and that is the way to get more cures for cancer. So I'm going to talk to you about, what is really now rapidly emerged as a new pillar for for cancer treatment, and that is activating the immune system. So we've seen this diagram before tonight. It's about these hallmarks of cancer. And so I'll talk to you about what really is a fascinating area of science, and that is the ability of cancers to hide themselves from the immune system. So cancers are clever. They can hide themselves from the immune system and not be rejected. But the good news is that now, in an increasing proportion of patients, we can turn that around and get the immune system to effectively reject the cancer. So this idea about the immune system and cancer has been around for a long time. So over 100 years, actually even older than the Walter and Eliza Hall Institute, where Paul Ehrlich made a very interesting prediction and said that if the host defences, our immune system, broke down, that you would get cancer with incredible frequency. And he actually got it right, because when we turn off the immune system in organ transplantation so that you can receive, a donor heart or a donor kidney and not reject it, well, you do get higher rates of cancer. So he was exactly right. But this concept of the immune system protecting us from cancer got a further big push along conceptually by Sir Macfarlane Burnet, who is a Nobel Prize laureate in physiology and medicine, former director of the Walter and Eliza Hall Institute. And in the 1950s, he really pushed the idea that accumulation of tumour cells in the body is stopped by the immune system rejecting those cells. So the effective immunological reaction against those tumour cells helps preventing us from getting cancer. Now, this is a really, really profound and important idea. And the reason for that is that if this is true, if our immune systems are stopping us all from getting a very high frequency of cancer early in life, then that means the cancers have hid themselves from the immune system. So the cancers have worked out a way of turning off the immune system. That's profoundly important, because if we can work out how to turn the immune system back on, you might be able to reject the cancers. So the path to doing that, and there is good news, I'll show you some data, some examples from patients a bit later about how the immune system can indeed reject established cancers. But to get to this point, we've needed science. And a lot of the fundamental scientific discovery here actually occurred at the Walter and Eliza Hall Institute by Professor Jacques Miller, who back in the late 1960s identified that actually there were two main types of immune cells called T cells and B cells. Fundamental scientific discovery. He also worked out what the function of this gland sitting in the top of our chest called the thymus is. People thought it was just a rudimentary appendix-like organ that we didn't need. Now, what Jacques worked out was that this was the factory for making T cells. And these T cells, as I'll get to, are fundamentally important in the body's ability to reject cancers, stop us from getting cancer, but also can now be utilised to treat and fight cancer. So this new pillar for cancer treatment is immune treatment. In the middle of this slide, you'll see that there's a cell with its little like tentacles coming out. That's a T cell discovered by Jacques Miller, attacking a cancer cell. And that is the fundamental way the immune system can fight cancer and control cancer. Up on the top of this slide, you'll see on the left-hand side, a tumour encasing the liver and the lung around the chest wall, and then after giving a patient immune treatment, that has improved markedly. This is a PET scan which is measuring glucose uptake. So immune treatments can have dramatic clinical responses. So how did we get to now the ability of harnessing the immune system to treat cancer? This really came down to fundamental scientific research, understanding how this T cell can be activated and can fight the cancer cell. And the fundamental concept here was the scientific discovery that there are brakes and accelerators on the immune system. So our immune systems would attack our normal tissues in our body if it wasn't for the brake that can turn off the immune system. But you also need to be able to turn the immune system on to fight a virus or a bacterial infection that we are all exposed to, or to fight cancer and stop ourselves from getting cancer. So to understand these brakes and accelerators required absolutely fundamental scientific discovery. Didn't start out with cancer at all, started out in just understanding the basic properties of the immune system, and there was identified a lot of molecules, key molecules that are the brakes on the immune system. So on the left-hand side of this slide is a part of the immune system that is involved in expanding the number of T cells. And then on the right-hand side of this slide is the fact that tumour cells are pretty clever. They have molecules that can turn off T cells. So even if you expand the number of T cells, left-hand side of the slide, and they see a cancer and they want to attack that cancer, the tumour cell's clever. It can turn on molecules that then turn off those T cells. And that is how the cancer can evade the immune system. What is absolutely remarkable, and I still have to sort of pinch myself and kick myself about this, it turns out that one molecule in all of that complex circuitry we heard about from Kate and we heard about from Jayesh and Kylie, one molecule turns out to be pretty important for the ability of the immune system to be turned off by a cancer, and that's a molecule called PD-1. So what we now have in Australia is what we're calling checkpoint antibodies. They're turning off the brakes on the immune system, the checkpoints that keep our immune system at bay, turn them on again to fight cancer. One's called Yervoy, it targets a molecule called CTLA-4, and one's called Keytruda, which targets a molecule PD-1, and there are other examples of these checkpoint treatments coming through. So we're getting quite good results with these treatments. So even something as difficult as cancer spread metastasised to the brain, shown here on the left, complete disappearance by activating the immune system, immune cells travelling into the brain and eradicating these tumours. Even large cancers, I mean, 10, 20 years ago, people thought the immune system would only ever work when the cancers are very small, but in fact, turning off these immune checkpoint pathways can even, as shown here, left to the right, eradicate very large tumours. Now, I'm going to show you some graphs, and this is sort of now, the technical stuff, but I think it's very important for our fundamental question tonight is that will there ever be a cure for cancer? So what this is patients with melanoma. And just remember melanoma, because it turns out melanoma responds very well to immune treatments, and I'll get to later why. But the disease of melanoma, even when it's advanced and spread, if you treat with a CTLA-4 antibody that turns on the immune system, can lead to very, very long-lasting responses even when melanoma is widely spread. So what this graph shows us, the proportion of patients alive is on the y-axis, and then across the x-axis is the number of months after treatment. And what you see is what we aim for in oncology, in cancer treatment, is a plateau on the curve, and that is beyond that 60-month mark there, 5 years. If you're alive, that becomes long-term, and we now have people with advanced melanoma surviving greater than 10 years with this treatment. But the second molecule that came along, this PD-1, is even more promising, and more patients are surviving a long time. But what's perhaps fairly obvious, but if you get a response, that is your tumours shrink with the immune treatment, then the survival curve shown on the top there in the green, patient survival is very, very good. It appears so far that 80% of people that get a tumour shrinkage with the PD-1 antibodies in melanoma will be long-lasting. However, if you don't get a response, the survival is shown there for patients with advanced melanoma in the black, which is actually pretty dismal. So a key thing is to understand why some patients respond and others don't. And you know, when I talk to my patients at the moment, still in 2015, when we give these immune treatments, it's a bit like rolling the dice. You know, your numbers might come up or they might not. And what we really need is the ability of getting so-called precision medicine and developing predictors of which immune treatment to give which patient, and that requires a lot more research. So how are we doing that? Well, you can sequence the genome. We heard about genomes earlier tonight. So you can sequence the genome of your tumours, and that may give you some useful information. Now, interestingly, I mentioned the disease of melanoma. So melanoma is the big mess circuit tumour. It has, if you count the number of mutations, this is a graph of many different types of cancer, way over there on the right is individual cases with melanoma. It is unfortunately the king of the number of mutations in the DNA of any cancer, but it responds best to the immune treatments. And we think that's because there are many mutant proteins that the immune system says, hey, you're foreign, I'm going to attack you, where tumours with less mutations don't respond as well. So using this new DNA sequencing technology, we can start to predict who may respond to these immune treatments. The other thing we're doing is looking at the types of cells inside the immune system. This requires sophisticated techniques to look at types of immune cells. And if you've got a lot of immune cells in your tumour, so shown here by the colour of the red and green, this tumour happens to have a lot of immune cells in it, and we're now knowing that if you've got a lot of immune cells in your tumour and then you use one of these new immune treatments, you're more likely to respond. But we need to do a lot more because we need some certainty for people, have the right immune treatment for the right patients, similar to what Jayesh talked to you about with targeted treatments. So there's been a lot of interest in the immune system now. It's been labelled the fifth way to fight cancer, and together with our traditional cancer treatments and these new targeted treatments you've heard about tonight, I think I'm very confident we are going to get more cures for cancer going forward. Thank you very much.
Thank you, Grant. I'm going to ask the other speakers to come up as well. So we've heard a lot of the complexities and challenges, but also some really exciting treatments. I think there's an opportunity now for you to ask questions of the panel. So there'll be 45 minutes. Just a reminder, we're recording this session. And we've got two helpers with microphones. If they can wave, there's Beck, there's one microphone, and right, yeah. There's
Good evening. Question to any of the panel. With miniature radiation, there's little microspheres I've read about in the press where they target cancer in the kidney, I think it's the prime one, and maybe the next door to it. Any comments on that? I think they're made by a company called Certex.
Yes, so there's been a bit reported recently because there was an important trial that was presented at a meeting in June this year in Chicago. So the concept is, and there are actually many different products that are available out there, but the one that you talk about is, the idea is that you deliver these little beads that contain radiation in them, and that radiation only travels a very short distance. So you can inject it via the blood vessels into a particular organ, and in fact the liver is the main organ in which this type of treatment is used. And the idea is that that treatment can be delivered there via the blood vessels. It disperses itself within the blood vessels, and often cancers have actually quite haphazard blood vessels, so it's easy for it to get into there. And that can be quite effective. So far, the information we have suggests that in that particular organ, so in the liver, it may help in delaying the time it takes the cancer to grow. But the problem always is that the only place a particular tumour is in. So often with cancers, they may not be confined to a particular organ. So delivering a local treatment to a cancer that is not just localised is often the challenge there. So I think at this point in time, watch this space, but there's certainly some interest there.
Other questions? to put your hand up high so we can see you. Maybe I'm going to ask Kate a question, going right back to the beginning, and really the causes of cancer. You've spoken about, we hear so much about smoking causing lung cancer, and yet people also say, well, my relative smoked for decades and has never developed cancer, or we meet somebody who's quite young who's never smoked and developed lung cancer. Are you getting any insights into why these differences?
Yeah, so that's a great question. So actually, smoking obviously is the largest cause of lung cancer. I think the statistics are about 85% of lung cancers are caused by smoking. So if you remember back to one of my original slides where, these smoking or different agents can cause damage in your DNA. So these can cause changes, and if these changes occur in genes that are quite critical for allowing a cell to continue growing, then you can imagine that in that individual, a cancer can result. But you could also imagine that these changes can occur in other places in your genome where there is no effect. So actually, the slide that Grant showed, next to melanoma, lung cancers were, I would say, the second and the third highest tumours that have high mutation rates. So obviously that's due to smoking, but maybe not all of those changes, are going to result in a tumour. So actually we would be interested in why that uncle did not develop lung cancer after smoking. And actually you raise another interesting point is, can you develop lung cancer if you don't smoke? And actually the levels of lung cancer in non-smokers is increasing, and actually more so in women, which is now around 15%. So I think the stats for Australia is that I think 1 in 5 women who present with lung cancer actually have not smoked. So we don't know why that is. We'd be interested, of course, in finding out, looking at the genetics on why that is. Are there environmental factors? So another statistic is that in China, the rates of lung cancer, especially in women, are extremely high. And this is thought to be due to the fact that women are at home cooking a lot of the times in coal type kitchens where maybe they're exposed to passive smoke. So these are some interesting statistics that are really now becoming really important in the lung cancer area.
Different sorts of environmental factors in different places.
Yeah.
Grant, is the same true of melanoma and sun exposure? We get different factors, or is it mostly sun exposure that is a factor there?
Yeah, so Australia has the highest rates with New Zealand of melanoma in the world, and that is due to the fact that we have a predominantly Northern European Caucasian population by our skin type, and we have very, very high levels of UV. In fact, I was just looking in the paper this morning, and in fact already now between 11:30 and 1 p.m. we have enough UV even here in a freezing cold Melbourne day that it can damage the DNA in your skin. We have so high UV in Australia. So melanoma high in Australia. It's caused by UV, what is interesting, getting back to this question about is it all due to UV or is it like lung cancer? Well, yes, you can get melanoma without UV. So Africans with very dark skin can get melanoma, predominantly on the soles of the feet, the palms of the hands, in areas that don't actually see very much sun. You can also get melanoma arising from your eye that never sees the UV. The UV radiation gets filtered out by the eye, and yet you can get melanomas at the back of the eye. And we know scientifically that it's not due to UV, because when you do the DNA sequencing that Kate and Jayesh spoke about, you don't get the telltale signs of the type of changes in the DNA that are unique to UV. So we can sequence a genome in a patient with melanoma and we know whether it was caused by UV or not. And there is this subset that are not caused by UV. But in Australia, it's only about 5 to 10% of all melanomas are not due to UV. So it's predominantly due to UV, the radiation from the sun or solariums, and therefore you can potentially prevent the disease, which is very important.
Any questions? There's a question down the front here.
Hi, I was just wondering, what role, if any, there is from a diet point of view and nutrition in improving the immune system, but I guess obviously there's a role in prevention, but also I guess in treatment of cancer as well.
Thank you. Becca's a question right at the front.
Yeah, so, diet is very important in cancer. So we know that certain diets predispose to the development of cancer, and that's where the best evidence is. So, bowel cancer, for example, in diets that are high in fat, low in fibre, you're more likely to get colon cancer. So there's, alcohol is associated with cancer. Breast cancer, perhaps, is a very common cancer, the most important one. So, what you take into your body is extremely important in the formation of cancer. What is less clear is how important is diet if you've already had an established cancer. So, many of my patients ask, well, what can I do with my diet to improve my outcome of having cancer? Well, we know that in now there's good evidence for both breast cancer and bowel cancer that if you're overweight, you have worse outcomes. And so actually, having a healthy body weight and watching what you eat is very important there. But can you improve the immune system with the diet? And that's a very common question. Well, really, there's no evidence that you can do that. I mean, what I sort of am very simple and pragmatic about it. I say to patients, follow the Cancer Council or the Heart Foundation diet. They're healthy diets. They are associated with low rates of getting cancer. We know that they certainly are not harmful in any way to the immune response. And, these watching your weight, your calorie intake and potentially low-fat diets, in some cancers you get lower rates of recurrence. So yes, there's plenty of things people can do, in a dietary sense to both reduce their chance of getting cancer and reducing the chance of it coming back for some cancers.
Thank you. The front here.
Thank you very much. And I have a couple of questions. So the first one is when you're talking about the PD-1 treatment, is it lifelong treatment or just when the cancer cell dies off and the treatment can stop and they will never come back? This is the first question. And second question is, I've been reading about the gene therapy, which is you can modify the gene in the T cell and then make the T cell become a killer cell and to cure a lot of terminal cancer, especially leukemia or something. It's any like a much more research done on that pathway?
Yeah, so look, I might start off and I might take the how long the PD-1, and then Kylie, why don't you take the really exciting data about CAR T cells and targeting, molecules on malignant leukemic cells is really sort of exciting new technology. So just on the PD-1 question, I'd love to know the answer to that because this is all new and it's come very quickly, and we don't actually know how long you need to keep the immune system, at bay, and we desperately need to do some clinical trials, and that requires actually a pretty challenging in some ways design of a clinical trial, and that is that you toss the coin and randomise half the patients to stop the PD-1 treatment and half to keep it going, and then look to see if the cancers will grow again in people that have responded. We think that's worth doing because there's lots of anecdotes of patients that have had to stop treatment with the PD-1 either for side effects or because they've got sick of it, because it's an intravenous injection every 2 or 3 weeks, and after a couple of years, that can wear you down. So some patients have stopped the treatment and there's been absolutely no regrowth of the tumours. So we need to do some scientific study to work out how long. We think it's not a short-term treatment though, like you give, 2 or 3 treatments over a month or two and then you're done. We think that, you need to keep the T cells attacking the tumour for at least a period of months. Exactly how long, though, we need to do trials to work out. Great question, really, on the, it's on our minds all the time at the moment. And Kylie, I'll get you to comment on those engineered T cells.
So the other really exciting thing with T cells is that whilst what Grant's been talking about in the PD-1 is when the T cell and the cancer cell are there next to each other, the cancer cell is evading that detection by the T cell. One of the biggest issues is the T cell actually finding the cancer cell in the first place. And so an area of work, particularly in leukaemias that there's been done, is engineering T cells, so these are the immune system cells, and putting on them essentially a marker, making it's sort of almost like a matchmaking service for cells. And so that the T cell then has a marker on it that says, I'm trying to find, the particular cancer cell, and we can be quite specific about this. So at the moment there are lots of trials in acute lymphoblastic leukemia, the leukemia that's very common in children. And essentially what it is, these engineered T cells now have a marker that links them in and connects them up with the specific cancer cell. And then the T cell can just do its job, which is kill the cancer cell. And this is really exciting therapy because, again, it's only been done at the moment in patients who have no other standard treatment options. So the trials are very early and they're only in patients who don't have other options, but the results have been quite amazing. And again, the side effects in the short term, because we don't have any long-term data with this, are very exciting. And we're often seeing this as being able to be used as a bridge to something like bone marrow transplantation. And all bone marrow transplantation is harnessing the immune system. It's introducing a new immune system, a donor immune system. And bone marrow transplantation largely works in blood cancers by giving the patient a new immune system and allowing that new immune system to attack any of the cancer cells that come up again. So it's just a more sophisticated way with hopefully less long-term side effects of harnessing that immune system to attack the cancer cell. Very exciting times.
So I might just follow up too with a comment there. So what's really intriguing to me is, and we heard, Kate talk about lung cancers have lots of mutations and melanomas have lots of mutations, and you give these immune treatments and the cancers can respond, is what do you do about the cancers at the other end of the spectrum that have hardly any mutations? So the immune system has a lot of trouble seeing them, right? Because they don't have all these funny looking proteins that are mutated. You're foreign, I'm going to attack you. So what can you do? Well, this is, I think, really exciting idea that we might be able to take the lung cancer in the non-smoker or the eye melanoma that has hardly any mutations and get these T cells and make those T cells, engineer them to attack those non-smoking lung cancer cells or those non-UV melanoma cells. And so I think that might be a really interesting technology for some of the hard-to-treat cancers.
Hi, thank you very much for the talk. I would like to have 2 questions. First, I would like to return to the diet. I read in a couple of articles that calorie restriction can improve or can prevent cancer. How reliable are these research data? And secondly, I would like to ask you, when I was doing research more in engineering, I read also a couple of articles and I went to the conferences, and there were some pretty promising therapy and research in using nanotechnology in cancer treatment. When they sent some, I think, gold particles, targeting research, so tumors, and they were pretty promising results. So can you please briefly talk about? Thank you.
Start with the nanotechnology.
Yeah, well, Jayesh, do you want to perhaps take the nano? You've done a bit of research yourself in that space. Yeah.
So look, nanotechnology is really interesting. So if we sort of take it back a couple of steps, the idea there is that there are some cancer cells where you know that there is potentially a target, right? And the problem, though, is that with these targeted drugs that we have at the moment, you simply can't get into that particular target, that protein, for example. There's a protein known as RAS, for example, which is quite complex in the way that it behaves. And therefore, using these targeted drugs, as I've talked about, you just can't simply get in there and target that directly. But there is an approach where you might actually be able to target the RNA that's related to this RAS protein, right? The problem with RNA is that it is unstable, so you can't get it into the cell. So one of the thoughts is, can we actually come up with a way of using nanotechnology? And essentially what it is you've got a molecule that you want to get into the cell in a particular way, but it's not going to survive getting to that point because the cell's got other defences in the way. And maybe you can come up with a way of packaging it, and there are lots and lots of different technologies that are being tried for that. Some of them are showing some early promise, and whether that's going to really, take it that step further, I think remains to be seen. Because fundamentally, the packaging, the nanotechnology that is contained needs to actually get to where it needs to get to at the end of the day. And often what happens is these things get consumed in the liver, for example, or by the immune system or something along those lines. So I think it's a space to watch. I think there are improvements that are being made, but they're not quite getting to the clinic in the way that we want them to. But clearly there's a lot of interest in that, and I think we will get there to some extent.
The first part of the question about diet, I think, goes to the sort of claims that we often see in newspapers. There are many things that come out about the latest risk factor or cure. Maybe you'd like to comment on that specifically, or generally about how you handle those sort of constant things that come up with this is the cure for cancer, or this is the latest risk factor, you shouldn't be eating peanut butter, or whatever it is.
It's really hard. I mean, and Grant touched on this in relation to, so there clearly is some understanding of the role that diet plays, and bowel cancer is a perfect example. So, a patient who lives in Japan, who then migrates to Hawaii, who then migrates to California, interestingly, their population risk of developing bowel cancer actually goes up in each of those steps. So there clearly are other factors that play a role in it. Maybe they're dietary, maybe they're environmental, maybe it's something else. The very direct question about, can dietary manipulation actually treat my cancer is one we do get asked quite often, and there are various, as you can imagine, there are various things that come and go. A sugar-free diet, for example, is one that's popular, and that's based on the concept that, by starving cancer cells in the laboratory, we find that these cells actually die. I think the difficulty I have is that cancers are essentially parasitic. They find ways of getting that carbohydrate in your system, and I don't think simply starving yourself of sugar is going to really be effective enough as a way of stopping those cancer cells. I think you'll find many other cells in your body actually being affected well before that actually happens. So I hope that answers your question.
I think, could I add, for the blood cancers that we treat, we use very, very aggressive multi-agent chemotherapy regimes. And the people who do best are actually the people who are well-nourished, not obese, but people who have a good, wholesome, standard diet. And in fact, we will artificially feed most of our patients because, in fact, they often will come in undernourished because the cancer itself has been parasitic and taken a lot of their nutrition. And so, in fact, we will, whilst we're treating patients for cancer, we will actually give them, appropriate nutrition. Because in the end, your ability for your body's T cells and B cells, the immune system to fight infection, your ability for your body to heal and repair is all dependent on provision of good protein and good fats and good nutrients. And so, in fact, the worst thing from my perspective is a form of starvation or restriction of diet while someone's having a cancer treatment, particularly for the blood cancers.
Yeah, and I think that applies for solid cancers as well. We see exactly the same thing. One really interesting thing, though, about the calorie restriction is there's some really fascinating scientific data not in patients but in model systems where you can really interrogate it precisely. Firstly, calorie restriction leads to increase in lifespan in basic organisms. Where you can precisely regulate the amount of calories, it actually increases lifespan, which is pretty interesting. The other thing from a cancer point of view, in terms of models of animals getting cancer, so it's very interesting that calorie restriction can prevent cancers that are driven by certain gene mutations, but not others. Right, so that's is pretty interesting. So it means that if you restrict your calories and you watch your weight, you can reduce your chance of getting some types of cancers, but not others, because if the gene mutations that we've heard about tonight are different, then the calorie restriction won't work. But look, I think in terms of if we think about the burden of cancer in our whole society. There's this obesity epidemic everyone talks about. We would reduce the burden of cancer substantially if, if everyone was, on average, lighter, less obese, yeah.
I think there's a question just a sec, and then another one.
I just have a question for Kylie about the protein used for cancer cells in blood cancer. How does it specifically target those cancer cells? Like, would they affect other cells around the site, or yeah?
No, so this is the protein that I talked about in my talk. That so it actually mimics a protein that's present normally in all of our cells, or a set of proteins. And these proteins essentially lock and engage into a groove onto the pro-life proteins, and they sort of essentially lock into this groove. And what the chemists have been able to do is engineer a protein, a drug that is effectively the same as the protein that locks in this groove. And by the drug locking in the groove, it means that it affects the action on those cells that normal protein in the cell would have done. And that's been quite a complex chemical discovery to get such a small protein into a drug. So what that actually does is it targets specifically the cancer cells, because cancer cells have too many of the pro-life proteins. It does in fact affect normal cells, but only to a very minimal degree, because the normal cells don't overexpress or have too much of these other proteins, and so its effect is very limited. There are definitely some side effects on other cells, and there definitely is some degree of effect on other cells, but not nearly to the degree that we see with many of the DNA damaging agents that are the traditional chemotherapy agents. So in fact, we see much more limited side effects on the other blood cells, for example, and also on the gut cells, which are the two main areas where we see a lot of toxicity from standard chemotherapy agents.
Thank you.
Good evening. It's been wonderful listening to you tonight about cancer research and the treatment. So it's great to have that opportunity to hear what you're talking about. I guess maybe this might be a comment rather than a question. My sister was diagnosed with lymphoma 2 years ago, and so I just, being a bit sneaky here and taking the opportunity to actually say it's a bit ironical, but today I went with my sister to visit Kylie, and who said, you have no evidence of lymphoma. So I wanted to actually thank you, Kylie, and say the care that you've given her this year has been extraordinary, and I'm perhaps acknowledging the human side of the treatment and the care that patients get as well. Everyone in the hospital, when I'm sitting in chemo with her, or in the hospital, or going through scans or whatever, maybe I'm turning this into a question now, but how important do you think that is, apart from it's fantastic having all the research that you do, but how important is it, the care and the human side of treating a cancer patient?
So thank you, but I think that that's the most important thing. In the end, as much as we can talk about personalised medicine and personalised treatments and clinical trials, in the end, the person standing in front of me as a clinician, and Grant and Jayesh, as clinicians, is a person who has a very complex life and family and friends and a history. And it's very, very important that each patient is treated as an individual, not just being their cancer as an individual cancer, but that particular patient, because so much of that weighs into their treatment and how they, how well or not well they deal with their treatment, the support they've got around them, everything from their nutritional status that we've touched on right through to their supports at home, whether they're a young mum with a young family, whether they're an older person who doesn't have much social support. All of those things are so important. But in the end, it's very, very important for us as doctors not to forget that the people that we deal with on a day-to-day basis, and it's perhaps the most rewarding part, or it probably is the most rewarding part of being a doctor treating patients with cancers, is being able to be part of their lives at a time when it's very, very challenging, and hopefully make a positive difference by providing them with information as well as hopefully treatments that will improve their outcomes. So it's a very, really the most important part, I think, of treating cancer.
Yeah, look, I think it is a really important point you make. I was an editor for a journal, and I accepted this paper in a disease that Jayesh treats, sarcoma, that showed that the survival for people with sarcoma that were single as opposed to married was significantly worse, right? And I think this is absolutely, so you better all go out and get married. But and now in Australia, of course, it doesn't matter what your sexual orientation is. We're heading towards marriage equality, I very much hope. But that's really getting at your point about support and what Kylie's saying about support. Data. This was data on thousands of patients. So the survival is better if you're married than single in the disease of sarcoma. Yeah.
Just to comment, so, I talked about something called personalised medicine, right? And the reality is, as a clinician, I think we all feel quite uncomfortable when we talk about that in the context of it being about technology and testing and sequencing and all of those things, because we all know that in reality, probably 90% of what we do is personalised medicine. It actually is, the assessments that we make, the perspective that we have, is so critically dependent on what's actually going on in our patient and in their lives and their support network around them and so on. So we use that term, and I think many of us as clinicians use that almost a little bit tongue-in-cheek to some extent, because the reality is personalised medicine has been around for a very, very long time. This just happens to be an aspect of it.
Thank you. We've got a number of people who want to ask questions. There is some order we will get to you. So at the back with Rebecca,
Good evening.
and then we'll come back down.
First of all, I'd like to thank the organisers for organising this, because it's an eye-opener. Professor Grant, some 11 years ago, he saw my husband. He's still alive. I'm here because I'm interested. 5 years ago, I was, again, it's a personal story here, breast cancer. Listening to everybody here, it seems as if there will be a cure for cancer. If there's a list, rather, we're hearing a lot about melanoma and the other diseases, but what about Kaposi sarcoma that is not AIDS-related? We don't hear a lot about that. Thank you.
So that was age-related?
Yeah, Kaposi sarcoma that's not due to AIDS. Yeah, so it's a rare type of a skin tumour, and it's one of the very interesting cancers that's actually due to a virus. Okay, now that doesn't mean that it's like the common cold and you catch it from being in the same room as someone. It's not contagious at all, but it's actually due to a virus. So we have a number of cancers now that we know are due to viruses. The most famous is cervical cancer. So Professor Ian Fraser, discovered the way of making vaccines for cervical cancer, and hopefully he's going to just, wipe out most of that disease in the next few decades worldwide, because you can take a vaccine to prevent infection from the virus. But there are other cancers as well, throat cancer. There's a type of skin cancer that we research called Merkel cell carcinoma. There's the Kaposi sarcoma that are not due to a loss of T cells, which is what happens in AIDS, but actually due to the virus. So, we're very interested actually as to whether the viral cancers might respond better to these immune treatments like PD-1. That's just a hypothesis at the moment. It needs to be tested in clinical trials. So, I'm hopeful that we will make significant advances in some of these virus-related cancers.
Got a question in the middle and then we'll go up the back.
This has been a fascinating session on advances in cancer treatment, and I thank you very much for it. But I'm interested in what happens for this information, how this information gets to the coalface, to the GPs, the specialists in the community. And I'm wondering if you could comment on any strategies that you're using to promulgate the concept of personalised cancer medicine in the broad community.
Jayesh?
Sure. Look, that's a really good question, and in thinking about it, I wish there was a simple answer, because the reality is, getting that information out is actually quite difficult. Look, obviously, fora like this help. There's I think actually quite a lot of publicity in the media and so on that informs people about what's going on with cancer. What some of the latest breakthroughs may be. There's been a lot about immunotherapies in the last year or so, which I think has obviously been really good. But how do we actually get that information directly to GPs and so on? Look, I think some of that is improving slowly. So even the universities, for example, are now getting actually more involved in thinking about, so the VCCC, for example, actually has, so there's thinking about Melbourne University and actually School of General Practice, there actually are very strong ties. And one of the important things that the VCCC has as part of its mandate is actually engaging with the community in that way. Not just in terms of passing on information, but also thinking about research as well, and clinical research and asking some of those important questions. I don't know if anyone else wants to comment.
Yeah, probably the other important things there that is becoming a focus now is health literacy in the community. So if the community at large, have more knowledge about health, then they're more able to engage with healthcare providers and get on the right path. And so that's, I think, very important, is a growing area of focus, so health literacy. The other thing that, I think is terrific in the cancer space now is the growth of advocacy and patient-driven support networks. And this is where social media and internet and everything can really have a great benefit to help people get on the right path. I mean, we do a lot of efforts to educate our colleagues, and we do a lot of effort in that, but I actually think it's going to be a partnership between improved health literacy, using a lot of our new information technologies, patient advocacy and support for patients for each other, as well as, our efforts in educating the healthcare workforce. So we need to do all of that, I reckon. Yeah.
Thank you. Okay, at the back.
Sort of question about clinical trials, because we rely on people to volunteer to participate in clinical trials, so the benefits of these discoveries are realised in patient care. And I was just interested in what your comments might be on what's the environment today like for conducting clinical trials, say, compared to 10 or 15 years ago, and what has been the impact, or what is the impact of personalised medicine on how you conduct those trials and in terms of their sophistication or their design?
So I'm happy to answer that. For clinical trials, so in fact, one of the benefits, and one of the biggest benefits of the VCCC is that the patient pool to do a clinical trial, and clinical trials are very costly, and they're one of the biggest inhibitors of doing clinical trials, particularly in Australia, when the Australian dollar, although it might be a bit better now, the dollar's not quite so good. But it has been a big inhibition for multinational drug companies to come to Australia, has been the distance involved, the cost of doing clinical trials here, and the relatively small patient population. And we're seeing that impact on our clinical trials more and more in Australia, in that the bigger clinical trials where they're looking to accrue many patients rapidly are no longer coming to Australia. And that's impacting us and our patients sometimes by us not having access to the latest drugs. And one of the things that clinical trials do, although they're very altruistic for the individual involved, is they potentially provide access to a new and exciting compound or drug. However, on the other hand, what Australia I think is doing very, very well and what has really changed is the focus from maybe 10 or 15 years ago on late phase clinical trials. So clinical trials, by that I mean trials of drugs that have been already tested in patients with that disease, and we know that they work, and we're really just trying to finesse the details, and they're large trials with large number of patients. Moving from those sort of big trials through to early phase 1 and 2 trials, which is where they're first in man trials or first in those patients with those drug trials. And Australia and Melbourne in particular have developed a real expertise because of the unique interactions. Clinicians such as myself and Jayesh and Grant, who are clinician scientists, we've all got PhDs, we've all spent time in the laboratory, and we're able to, and there's more and more doctors these days have that background, able to provide that dual focus across science and medicine. And these early clinical trials really need that background because it's really taking the drug into the patient and looking in a very detailed way at how that patient not only tolerates the drug, but why that patient responds to the drug when another patient may not. What markers have we got, for example? And that's what's really changed, is the level of detail in the clinical trials that we're doing. The clinical trials are smaller, but that's also an aspect of the breakdown of the diseases into many smaller components. So no longer do we have one trial for leukaemia, we have 7 or 8 trials for all the different subtypes of leukaemia. So the trials are smaller, they're more specific, with a lot more detail in them, which is enabling our patients to access sort of more detailed treatments.
We've got time for 1, maybe 2 more questions, and there are a number of hands. There's a gentleman in the middle that's been waiting for a while.
Yeah, thank you. Thanks to the panel for a really stimulating and informative evening. I'm still drawn in by that provocative question. Will there be a cure for cancer? It seems like a crazy question to ask, given that it's not one disease. But we've had this lovely breakthrough recently, these breakthroughs recently with immunotherapy, so anti-CTLA-4 and the PD-1s. And suddenly there is hope that there are therapies out there that have very broad application. And I just wonder whether or not the panel has a view on where the larger transformational breakthroughs are going to come from in the near future. I know a very large question, but I am interested to know.
So in some ways, the question is, which one of you is going to win the Nobel Prize? But no, I love it. I love the question. So, I mean, you're absolutely right that we make some big advances through transformative, very original science, and then on the other side, we actually, in terms of total numbers of people that are going to be cured from cancer, we make these little incremental steps. So we've got to do both. We absolutely got to do both. We've got to invest in people with, way out ideas that might just really transform things, as well as make sure that, we can make those little incremental steps with ideas that are already there. So which are the big, next transformational things that are going to happen? I mean, maybe it's understanding, I'm saying this to the panel and then I'll explain it. It could be, the non-DNA, the epigenome, right? The fact that actually, we heard about how our genes make proteins. Well, that is actually only about just less than 2% of all of the DNA in our cells. There's all these areas of our genome that don't make any proteins, and there's also all these proteins that are wrapped around the DNA that determine whether genes are on or off. And we're starting to understand that that's important for cancer. So one really, I think, cool experiment actually is you can take a cancer cell and you can put it into some situations in an animal, and it'll turn into a more normal cell again, even though the DNA hasn't changed, right? So I'm having a bit of a guess here, because otherwise I'd be given a Nobel Prize, but the next transformation I just wonder might be moving beyond the DNA. I mean, we're going to make lots of advances now with all the DNA stuff we talked about tonight, but anyway, that's a guess. That other guys may have some far more clever ideas than me. Yeah.
I think one of the biggest challenges is actually going to there's almost an avalanche of new therapies coming through, and from the immunotherapies to the monoclonal antibodies, which we haven't really touched on, but target cells specifically to the CAR T cells, enormous or cell death pathways. It's going to be, I don't think any one single treatment is the answer, and I think the breakthroughs are going to come by the rational combination of some of these therapies together and working out which ones go together at which time and sequencing. And I think that is going to be different for every different cancer. But I think that's going to be one of the key things is being able to, and also having the ability to have the science plus the means to combine them rationally and work out the difference rather than one particular breakthrough. It's going to be that the person who can combine those in a non-toxic and effective form.
Yeah, I think, I was thinking very much in the way that, Kylie has just expressed. And I think for me, the transformative side of this is it's almost sort of turning the question on its head, which is not so much transforming what we necessarily do with a cancer, but it's almost transforming the way in which we go about it. I think that's probably going to be the key. We've all talked about complexity, and we've all talked about, understanding the sort of depth that's required to be able to really make a change in that way. Now, you get lucky from time to time, and we'll come up with simple breakthroughs along the way. But the reality is, how do we actually put all of this together? How do we have a health and research system, right, that is actually going to allow us to do that in a very real world setting, because this is very real world, as we all know. And that, I think, is probably the transformative part of it. And look, we're all excited because we work within an environment where we actually get to do that to some extent, right? And, that occasionally has a real impact in a particular patient. But the question is, how do we keep doing that? How do we take that next step further? And for me, I think that's probably the critical, a critical part of it.
Yeah, if I can reflect on lung cancer, I think what I didn't show tonight is actually the survival rates for lung cancer are really the worst of all cancers. And I think that's probably the biggest hurdle that we need to overcome if we're really going to conquer this disease. And probably a way, there's been some ideas thrown around about how we can do that. And I think one important way is trying to find the cancer early. And in breast cancer, they do this really great. So they have the mammography and, survival rates for breast cancer are over 80% now because they have these early screening detection. So for lung cancer, there's been, a few screening ideas thrown about the space. But I think, that's a way that I think we can really tackle the cancer just before it gets, starts to overtake. We can really attack it at this early stage. And I think that's going to be an important area.
So there could be something in the blood, right, Kate, that, a blood test or some transformative thing like that, some technology that, you can detect cancers in the blood. I mean, we're a long way away from that right now, but it would be transformative, wouldn't it? Yeah.
So that's probably a good question. I realise that there's many more people who have questions. I'm sorry that we're out of time. I think that's probably a very sort of optimistic, in a sense, note to finish off on. Well, there's recognition this is a very complex problem, and there's probably not going to be a single cure. We are in a period of transformation, and there's huge optimism and possibility and amazing research going on in Melbourne. I think it's really evident from this discussion today that these 4 researchers and clinicians here are really world leaders in their field, and we're very fortunate from the work that they do and also the work the institutes and hospitals are doing in Melbourne so that patients in Victoria are really benefiting. So I'd like to thank very much the 4 speakers, Professor Grant McArthur, Dr. Kylie Mason, Dr. Jayesh Desai and Dr. Kate Sutherland for their time and contribution to this discussion today. I'd also like to thank our event partners for this evening, Federation Square and The Age, for their support of tonight's event, and to the Walter and Eliza Hall Institute for hosting the event, and happy birthday. And thank you all for attending. I hope that this has proved sort of fruitful and interesting for you, and that you're taking something away from this about the future and hope for the future of cancer, and recognising that wonderful work that's happening in Melbourne and outcomes. Thank you.